So my name is Georgios Pappas. I work at the University of Brasilia, and I'm going to present to you some challenges and perspectives of bioinformatics teaching here in Brazil. Okay, so first of all, Brazil, I'm really far away from everybody. We're located here in Brasilia, the capital of Brazil, far away from everything. And Brasilia is a nice city with very modern architecture. So I'm going to start off Dr. Rhodes, giving you my personal experience in bioinformatics, even before bioinformatics was born. I'm not that old, but I'm from the 80s, you see? So the 80s was really nice. And in the 80s, I started programming with computers, so to speak. Okay, so what we had was this machine with 16 KB RAM. We didn't have monitors, we didn't have hard disks, floppy disks, anything else. But it was really fun having being initiated in, let's say, computers even before college. Okay, but in the 80s, there were several things missing. Okay, first of all, Linux, several programming languages that we use today, like Python, Java, R, World Wide Web. We didn't have this. We didn't have cell phones, GPS, MP3s, and so on. But it was really fun. So moving to the university, since computer science was such a new field, my other passion was biology. And of course, I got a formal education in molecular biology. So undergrad degrees, a degree I got at the University of Brasilia, and I have training in all wet lab techniques to characterize biomolecules, anyone, DNA, RNA, proteins, okay? And even by that time, I did Sanger sequencing by hand, okay, running really big gels, like a meter-long gels with radioactively labeled DDNTPs. And in recollection, the wet lab was a very essential experience for me in my formation, mainly to get the notion of the complexity of biological systems. Okay, so, but exactly at that time, as you can see here in 87, I started programming and doing research at a lab that worked with protein secondary structure prediction, okay, based on amino acid sequences. And by that time, I started learning basic. Myself, I learned C, and I improved in terms of hardware, and I got this computer with 48 KB RAM, okay? But it was really fun. Then I moved to Illinois, University of Illinois at Urbana-Champaign, where I exclusively did work in computational biology. And for that, I was working with a protein structure, trying to categorize structural motifs. And by then, everything was programming, computers, and it was really nice. And my computational biology, this is very important. And it was in the 90s. And at that time, in fact, I was one of the first persons in the world to use a web browser, because it was developed like next door to me, the Mosaic web browser. And I recall we had like 15 links worldwide. That was all we got in terms of websites. And in my evaluation of the web browser, I said, oh, okay, no, this won't work. Bad decision. But okay, since last century, I never set foot in a wet lab anymore. I was strictly a computational biologist. So in recap, what I can say, I can say that I had a biological background from theory to practice, and continues computational to use in terms of programming languages, operating systems, algorithms. But this is important. No formal academic training. It was all by myself, reading through paper books, okay? And what happened? In the 80s and 90s, in terms of biology, we didn't also have many things, such as automated DNA sequencers, genomics, a holistic view of biology. You see, we had alternatives, so gel-based summary sequencing. Genomics was only in dreams, okay? And biology at that time, last century, wasn't what I can say holistic. It was, we had an individualized view of individual components. Oh, this protein, this promoter, and that's it. And what we didn't have was this term, bioinformatics. We used to have computational biology. We still have computational biology. And everything we did was computational biology. And then, of course, the genome era of biology began. And it was like the big data era of biology. What happens is, let's say it started at the end of last century, as all of you know, in 95, with this first genome published. But then in the 21st century, we have explosion of genomics. And this is a new trend, a new paradigm for biology itself. So at least to begin with understanding complex organisms, we need to have the DNA blueprint of the organisms in order to try to relate phenotypes with genotypes and several other functional aspects of biology. And this is going on, okay? So we even have this very interesting project, the Earth Biogenome Project, in which they want to sequence everything in the world. And this will be some very doable in the next decades, okay? Meaning that we are gonna have lots of data, let alone the data that we already have, okay? So I really like this paper here that I want to share with you, Teresa Atwood, from the beginning of the century, where they say that bioinformatics is immersed in a field of high throughput techniques that generate vast amount of data, but data can only be useful if we can turn in data into knowledge and knowledge into understanding. So I'm going to highlight the key words of what I'm saying: data, knowledge, understanding. Okay, and of course, what we have is when we are in the high throughput, not just genomics, all the omics techniques that generate vast amount of data. Of course, the new biologist should be inserted in this new reality. And the biologist should be proficient in doing some computational analysis. On the other hand, we have bioinformaticians exclusively linked to this field. And in this case, okay, excel in computational analysis of data. Why? We have to turn these data analysis into knowledge. But there's a big problem here. There are differences between these two profiles in terms of overall goals, the perspectives of what I can do with the data, and of course, in the training level, okay? And to turn data into knowledge, okay, this new breed of professionals have to know algorithms, other areas, statistics, aesthetics, as Dr. Rhodes told about how to present the data. Oh, this is very, very important also. But also we need biology. And to transform knowledge into understanding, here comes our focus, is biology. Okay? And with that, I should quote Wally Gilbert here, in which he says that biologists must be computer literate. And this is a new paradigm in the whole biology, okay? And in this new biology, for the 21st century, the author say that the new biologist should not be only a scientist that knows little about several things. We need a deep knowledge of one discipline, let's say biology, one of the biologists. Okay, we have several biologies, okay, and we need working fluency in several other sub areas, okay, and what are the challenges of teaching bioinformatics? So I've been teaching bioinformatics courses for 20 years, for the past 20 years, since 2001. And after 2 decades, I'm still working on it, okay? It's not straightforward. Why? Because it is very difficult to create and adapt a curriculum that will fit the needs of all student aspirations. Okay, this is there's a big diversity in this aspect here. But anyway, I divided all my courses based on student background, okay? And essentially, I have courses for undergraduates, graduate students, and for students interested in a bioinformatics career path. So I organize the courses like this with the for the background, the audience. So my audience is biology and biotechnology majors, and in fact, biological sciences, even agronomy, some medical students, and so on, and so forth. And the objectives here are just to provide a general introduction of bioinformatics and bioinformatics tools. And then we have courses for graduate students where the objectives are reflect the students seeking proficiency in using computational tools to analyze their data. And the final thing are for research. And of course, again, for only those who want to become bioinformaticians, okay? And so for undergraduate level, it's a single course, a 60-hour course. It's a required course with theory and practice. Per semester, I have about 40 students. And now, in terms of graduate courses, I teach multiple courses, and I will get into this later. And for research, it's personal mentoring. So I was happy to see that ISCB developed, let's say, what a bioinformatics core competence would be for several levels. So they had competencies for a bioinformatics user, a scientist, and an engineer. And this is, if I don't expect you to read that, but this is exactly what I have here. So I divided into tiers. So at the undergraduate level, the major focus is given to study biological databases, okay? And why is that? In which databases? Normal literature, nucleotides, proteins, everything related to proteins in terms of sequence, domains, enzymes, biological pathways, genomes, variations in phenotype, and data quality assessment of the databases, and of course optimized querying the data, and of course some basic tools. Everything is online here for this course, okay? And why this operation? Basically because of, again, Walter Gilbert, when he said, oh, a new paradigm in biology emerged in that biological data. And for that, the starting point of biological investigation will be theoretical. And with that, I can present you a simple, let's say simple, in 4 lines, a question that can be asked. let's say, how do I know all the protein coding genes from DNA repair system of the fungus C. neoformans? This is a very complex biological question. And of course, you can answer with using bioinformatics tools, but just doing canonical database surfing and filtering may not be the answer, you see. So we have some, what I consider optimized alternatives to using and querying that databases, and which it's kind of really simple. Okay, it's stuck here. Just a second. Okay, I'm having a problem with this slide here, but essentially it's gene ontology, ensemble, and biomark. Okay, but here we go. Gene ontology, ensemble, and biomark, and we can do this in less than 15 minutes. Okay, so this is one of the goals. So in terms of the overall appraisal of the students, this discipline has had very good reviews from the students, because they see bioinformatics tools as essential for their careers, even though they might not be bioinformatics. Negative points are the lack of textbooks. I'm writing one for them, and in but for my perspective, when they are doing studies and using the concepts given, the basic concepts given in bioinformatics. The negative aspect is that I'm having a relatively low number of students actually engaging in bioinformatics research. This is low, it is low. At the graduate level, I have one market division of courses. Okay, so what happens is that I teach several different classes, reaching NGS, Introduction to Linux, R programming for bioinformatics, okay, and pipelines for bioinformatics. I'm not going into the details here, but what are, in my perspective, what they need to know. First of all, NGS, they need to know the data. Linux, it's the work environment for computational analysis. Programming in R, for instance, it's our work tool. And pipelines in bioinformatics, it's our workbench where I can mix and match several bioinformatics programs to produce complex analytical workflows. Okay. I had some decisions. Okay, you might ask, why you were teaching R instead of Python? For me, since the students are only coming from biology background, I think R, it's a little bit easier to learn in like a 40-hour course, 45-hour course, okay? And in terms of pipelines for bioinformatics, the major goals were students, since the background is just biology, they should be able to install and run published pipelines. And what happened was that I was trying to teach them how to program in Nextflow or SnakeMake, but the total lack of programming skills wasn't a nice experience. So we had to halt this thing. Okay, so here I come with an example of the richness of how just simple,
I'm so sorry to
let's say,
interrupt.
Linux. Yes.
Oh, Dr. Pappas, we are running low on time. Do you think you could wrap it up in the next few
Okay.
minutes?
So
Thank you.
I'm just gonna skip this and for the research level bioinformatics, what happened? I need personalized mentoring. Okay, so we work with several plant genomes. We published this eucalyptus, the common bean genome, and we are working now with Brazil nut tree genome. But we also work in my group with several other species. With bacteria, fungi, with all omics technologies and all NGS technologies is a showcase. And it's very difficult to adapt to these technical changes. And this poses both opportunities and challenges regarding bioinformatics education. And so essentially, we have been developing several pipelines which need several tools, individual tools. So Nextflow, Docker, Shiny, R, and this is not trivial to teach in regular courses along the way. So just to finish up, my experiences were in terms of gender imbalance for graduate and undergraduate courses, absolutely no imbalance. But for research, I still observe a gender imbalance. And let's not forget that the mother and father of bioinformatics is Margaret Dayhoff. So we should close this gap. And just to finish, the new biologist needs several skills and working fluency in programming, statistics, and knowing the data, but deep knowledge into the biology is, let's say, what I consider the most important thing in the moment. Okay, with that, I'm going to skip the other slides, and I will finish up my presentation here with that. Okay.
Great. Thank you so much, Dr. Pappas. That was a very awesome talk. No worries. Okay, so we have a few questions for you. The first one is, where do you think bioinformatics is going next?
Oh, this is a very interesting question because since, let's say, I saw it growing from the beginning, and technology simply is molding, is shaping bioinformatics. So we started with, okay, we cannot have genomes, we cannot sequence DNA, then we can sequence short, then we can sequence long, and then bioinformatics, and this is a very important point, bioinformatics will change next semester. You see, we are tightly linked to technological advances, new biological discoveries, and bioinformatics is involved into that. So it's an ever-changing field. It is challenging, but it's really nice. Why? Everybody needs bioinformaticians. Everybody.
Great. Thank you so much. I think we have time for one more question. What are some bioinformatics careers that are available in Brazil specifically?
Okay, so in Brazil, we have a huge problems now. And unfortunately, career-wise, not for bioinformatics, Brazil should be an exception in the world. It's not because we are short in bioinformatics careers, we are short in science careers because economic and political difficulties. And what we're doing with bioinformatics, all my students, they are moving abroad. So all my PhD students are moving abroad. One is now in University of Florida and the other in Denmark. We cannot, Brazil cannot absorb. And this is detrimental for future development of the country, unfortunately.
Okay, well, again, thank you so much. And that was an awesome presentation.
Great. Thank you so much.